So we are going to start the third and last session on From Darwin to Evo-Devo. And it's a pleasure to have here Markus Affolter from the University of Basel, who is going to tell us, can we have his talk, please? He's going to tell us about branching morphogenesis in flies and fish.
Thank you very much. Thank you very much. I would like to thank the organizers for giving me the occasion. Okay. I'd like to thank the organizers for giving me the occasion to come here. It's my first time to Israel, and I usually go back with a plane I actually intended to, but this time they convinced me to stay a little bit longer, so I canceled the lecture. Something that I find is really bad to do. You cancel a lecture, your students will be probably happy. But so what I would like to do is probably From Darwin to Evo-Devo. It's probably more From Darwin to Evo-Devo to Devo. My talk will be mostly developmental. But obviously with some implications. I will talk about branching morphogenesis, and it is quite clear that from early on, branched organs have fascinated biologists throughout many, many years. The intricate branching patterns of the lung or the kidney, the mammary gland, tracheal system, and vascular system as shown here, and they're obviously quite. There's some questions that you can ask when you look at these things, and this is how are these branches formed, how are these patterns, how do they arise, how are they controlled, how are these stochastic processes or are these heavily genetically controlled processes? Obviously what we would like to know nowadays is what are the underlying cell behaviors. If for branched organs are these underlying cell behaviors the same? What are the genetic networks that lead to the formation of these branched networks? Do they control the same cell behavior? So is there some common theme there? And this is obviously then manifested in the question how similar are these branched organs at any of these levels here. For many years we have been studying the tracheal system in the fly embryo. We have now moved about 5 years ago to try to address this question of similarity by looking at blood vessel development in the zebrafish. So the question that we wanted to particularly ask from the very beginning on, and none of these questions had been previously addressed in any of the branched organs because this was early days, like 15 years ago. The questions that we wanted to address is what do individual cells do? Because cells are the building blocks. I think if you don't have an understanding at the cellular level, it is very difficult to to infer similarities and so on. So we wanted to understand and characterize what single cells do. How do they use signaling systems? What are the effectors of these signaling systems? What forces alter tissue architecture? Because very often when we talk about morphogenesis, we also must deal with forces, and this is not so easy using simple genetic tools. And then obviously we wanted to ask whether there are similarities or differences. The tracheal system of the fly embryo is obviously it's a nicely branched organ, but it's a very simple system. It starts off from a simple flat epithelial sheet. Cells are set aside by the expression of specific transcription factors. The black cells undergo branching, the white cells don't. These cells then first invaginate in groups of about 80 tracheal cells. And these invaginations, which actually already generate the lumen and generate sort of the initial network, these sacs then elaborate in different directions, branches form, and eventually these individually formed networks have to fuse to give rise to a functional network. Strikingly, when we started to analyze it was clear that from the invaginated sac-like structure to this extended network structure, there was no cell division. So it was rather clear that we could analyze this morphological process genetically, and what we would find is not differences in cell division rates and things, but we would find genes that modify the morphology in the absence of cell division. So this was an advantage. Obviously, other branched organs might do very differently. The tracheal system consists not of similar branches in the different directions. 6 branches are generated per unit, and these branches have different cellular architectures, and they represent all the types of tubes you can generate in nature. Some of the tubes are multicellular. Here is a junctional marker where you can see this multicellular aspect. Some of the tubes, and most of the tubes actually, beside the main tube here, are tubes made by single cells wrapped around the lumen, sealed with autocellular junctions in a sort of cord-like manner, head-to-tail-like manner. So this is a cell, a ring where the lumen goes through. This is another cell. So virtually all these branches here are made up by cells in this particular arrangement. And then at the tip, at the periphery of the tracheal system, individual terminal cells generate branches within cells. These cells generate tree-like structures, so they make hundreds of ramified branches, generate tubes within each of these branches that then ultimately connect up to these extracellular tubes. So at the periphery of the system, you have hundreds of individual tubes within cells that then connect up to cells, to lumen in between cells. It turned out that during this genetic and cellular analysis that there are four major processes, cellular processes that are involved in generating the tracheal system. Cell migration is a key issue I will talk about. Cell rearrangement has to accompany this cell migration in order to provide for these different branches here. Branch fusion is important to fuse these adjacent metamers. Lumen formation, lumen size control, and so on are also issues that I will not address here. So when you work on a problem like this in flies, what you obviously do is try to isolate mutants, do saturating screens, zygotic saturation screens, hope to identify those genes that actually lead to the branching process. These are 3 mutations with the same phenotype in 3 different genes. In each case, tracheal cells have been specified, but the branching process has been completely abrupted. So these are certainly genes that control the branching process. It turned out that one of the genes codes for a branchless ligand, mainly worked on and characterized by Mark Krasnau's group. Another mutant disrupted a gene which codes for an FGF receptor, and this was work mostly done by Benishilo-'s lab. A third gene mutated an FGF receptor-specific substrate. That was work by our lab and Maria Leptin's lab. So clearly an FGF signaling pathway seems to control this branching process, and sort of the existing evidence, or the evidence for this is now using ectopic expression pattern mapping, and so on, is the following. The FGF ligand is secreted by non-tracheal cells in the vicinity of these tracheal invaginations, of these tracheal sacs. So there are 6 spots of FGF expression. Branches grow more or less towards these 6 spots. So the expression pattern of FGF dictates the formation of branches. FGF receptor is expressed in the tracheal cells, and so is this FGF-specific signaling component. Now, from early on, when we started to tracheal development, we wanted to take a live imaging approach, because it's simple to say, but seeing is believing. If you watch a football game and France wins or ties 1-1, you don't know whether this was a regular goal or not. It's only seeing is believing. So this holds, of course, also for biology. And what we did from early on is to express, using tracheal-specific lines, a number of different proteins. This is actin GFP, and you can see that the tip cells here, 2 or 3 cells at the tip, migrate eventually towards the dorsal side. During that process, stalk cells reorganize to provide the lengthening of this stalk here. And based on these studies and these genetic studies, sort of the prevailing view is now that FGF signaling controls the branching process. It is read out by the basal side of these epithelial cells. Cells migrate, starting off from their basal side towards these FGF sources, and during the outgrowing process, cells rearrange behind to give rise to a stalk. Now, when you think about this is the end result of the branching process. The beginning is a flat epithelial sheet. So you can easily imagine that from this flat epithelial sheet to come to these very highly stereotyped branches, you also need extensive cell rearrangements, because in the epithelium from which we start, cells usually have 6 neighbors, and when we end the process, cells usually have only 2 neighbors. And these are cells wrapped around the lumen, linked to other cells on either side. So there must be extensive loss of cell-cell contact remodeling of an adherence junction. So this is sort of one of the basic inherent feature of the branching morphogenesis process. Now, when you look at these, so we were really interested in trying to understand this at the cellular level. If you look at these movies here with actin GFP, you don't see where a cell ends or where a cell starts. You don't see how these cells intercalate. You could watch this movie many, many times. There's no way you can identify the underlying cell behavior. It is a bit better when you start to look at these cell rearrangements using junctional markers, because these are epithelial cells. Every epithelial cell has an adherence junction border which defines sort of its shape, at least in the subapical region. So here you could start to see lines and rings and so on. But even if you look at this movie, it would be very difficult to really sort out what cells are actually doing. So several students in my lab, they started to label different aspects of this tracheal system. This is, again, a junctional marker using alpha-catenin GFP. These are nuclear markers here. And then doing a number of high-resolution movies allowed us actually to sort of get a model of how this intercalation or the basics of this branching morphogenesis process. So these are now still pictures of such movies with very high resolution. You see sort of two lines and then something complicated, two lines. These are two cells that sort of pair with each other, then another two cells pair, and another two cells pair. During the process, these cells intercalate, and the intercalation process sort of reads out from these junctional rearrangements. It's not easy to follow that, so obviously what you do is you color individual cells. So you have a green cell paired with a red cell, and then you just follow the colors as it goes, and from this you try to derive a model of how cells behave. The model that we derived from this type of artificial coloring was a model that is shown here. So the cells first pair, and then individual cells reach around the lumen until they meet their own apical junctions on the other side. So they make autocellular junction. Then the cells slip along each other in a process we call zipping, and then the whole process needs to terminate, because if you interconvert these intercellular junctions to autocellular junction, to the very end, the red cell will unlink from the green cell, and the whole process was for nothing. You don't change, you don't generate an interconnected network. So this is a model, and the student who came after we published this model, or after we had this model, said we really should try to look and label single cells to confirm this model, because the model is good, but you need to confirm it to some extent. So what he tried to do is to label single cells and to label the junction of individual cells. This you can do with a FRT flip system in flies. And what he saw then is indeed that in a non-sort of intuitive fashion, the junctional arrangement in live embryos goes from an open structure to a loop, line, big loop structure to two small loops and a large line, as we would have predicted if we would have labeled, when we labeled single cells here in a schematic fashion. So we think that this model is correct. Cells, in order to give nicely structured branches, some cells pair, they reach around the lumen. You have a zipping process and then a termination process. And obviously a model like this asks for a lot of other questions. How are junctions remodeled when intercellular junctions turn into autocellular junctions? Who provides the force for this? How is the process stopped? How is it developmentally regulated? Because some branches, there is no intercalation, and so on. So before I would like to go to fish now, I would like to give you some answers, which then will sort of provide the background for the second part of the talk. So what we know now is about the branching process in fly embryos is that there is a key molecule at the top of the hierarchy of the branching process, which is an FGF ligand, which presumably is secreted from these cells and establishes a kind of a morphogenetic gradient along which these branches then form. FGF so provides the migratory input, makes these cells motile. It provides also the forces we know now for cells to intercalate. It also segregates tip cells from stalk cells by the induction of delta in these tip cells, which then inhibits these migratory properties in the stalk cells. And later on, FGF signaling is also required to generate specific cell types at the periphery of the systems, these terminal cells that then further branch intracellularly. So the branching, the key molecule of the branching process is this FGF ligand, and Mark Krasnow at some point, he termed this, he coined branchless, or the branchless FGF gene, genomorph, because it codes for a protein that clearly gives, controls the three-dimensional structure of a given organ. This term has never made it into literature. It is not used anymore. One can discuss how significant or how, whether this would be a useful term or not. But clearly branchless controls not only migration, it controls tip-stalk segregation, intercalation, and further terminal differentiation. So it's not like the branching process would be controlled by many, many different things. There is something very inherent to the process of how you generate these branches. There are other proteins that do impact on this pathway. So not all the branches, this process takes place, this intercalation process. Many signaling systems control the expression of specific transcription factors that block the process. We don't know what the target genes here are. There are junctional proteins that are very important for this remodeling process of intercellular junctions into autocellular junctions. There are luminal proteins secreted apically by these tracheal cells that are very important to keep the cells together so that the system doesn't break, so for these termination processes. So as we were trying to characterize this system using live imaging, genetics, reverse genetics, and biochemistry more and more, we were, of course, tempted more and more to ask, what about these luminal proteins? Are they important in other branched organs? Are they really necessary and inherent part of branching organs? What about junctional remodeling? And what about epithelial remodeling via transcription factors and so on? So at some point we decided maybe we should look at similarities, and we were sort of tempted to do that ourselves as well. Since from all of these studies, I think the take-home message for us was that we can only assign functions to mutants and wild-type genes if we look at live imaging. Seeing what cells actually do allows us to really attribute important roles at the cellular level. So what we wanted to do is to choose an organism where imaging can be done very well. And that was quite obvious that we would go to the zebrafish. We decided then to go to the angiogenesis process, which might or might not be similarly controlled at the cellular level, ask exactly the same questions. What do individual cells do? How are they controlled by signaling systems, effectors, forces, and so on? The system we chose to analyze is the process called angiogenesis. So the first angiogenesis process that occurs in the early embryo. In the early zebrafish embryo, the vasculature is first established via a process called vasculogenesis, which then provides a simple circulatory system. And upon these systems, many branches are added by a process called angiogenesis. This is very similar to branching morphogenesis because in angiogenesis, these branches grow out of pre-existing branches. So they need these pre-existing branches. You don't necessarily generate lumen tubes de novo. You start them from pre-existing branches. Now, the system has been analyzed quite extensively also by Weinstein's group and by Mark Fishman's group. And here is sort of a cellular or a model of how these intersegmental vessels arise. This is the dorsal aorta, established by vasculogenesis. Branches grow into dorsal directions. This is shown here. You have very nice tip cells with philopodia that explore the environment. These branches grow out then towards the dorsal side. On the dorsal side, they change direction and spread out along the anterior-posterior axis until this specific structure is made here. As you can see, this structure only links artery with artery, so blood flow cannot really kick in, and you don't have connections between the arterial vessels and the venous vessels. Sprouting, this is called the primary sprouting. In the secondary sprouting process, sprouting occurs from the venal tissues, very similar to primary sprouting, but starting from the caudal vein. Some of these sprouts then connect to primary sprouts, and upon pruning here and fusion processes, the first connections between the artery and the vein are actually established. So there is a very small micro climate here, more or less, where all the features of branching morphogenesis can be visualized. Cell migration, cell division, cell rearrangement, branch fusion, lumen formation, pruning, and so on. And this is a movie that shows these intersegmental vessels. So what you can see here is that it looks actually quite similar. This is a low-resolution movie, but it looks quite similar to what we can see in tracheal development. We have branches that grow dorsally here. You can see how some of these branches then fuse. So here these branches grow. Here you can see, maybe not so well visible, how they explore the environment. Eventually they meet each other and grow along the anterior-posterior axis. The lumen starts now to be opened up here, and the lumen then opens up through the entire structure here. But still, these are still connections between the dorsal aorta and the dorsal aorta. Now you see very fine extensions from these secondary sprouts. You can see one here now that explores the environment. It finds one of these primary sprouts, makes contact with it, and the lumen starts now to be established along this side until the lumen is opened up to the vein, and then the blood can start to flow from the dorsal aorta through these intersegmental vessels to the to the caudal vein. So it's a very small area where you can study these branching processes. It turns out that the cellular architecture of these intersegmental vessels is even more simple than the cellular architecture of these dorsal branches we look at in the trachea. These are supposed to consist of only 3 cells: a T cell, a linking cell, and an inverted T cell. And in a later study by Brand Weinstein's lab, it was proposed that the lumen in these ISVs is formed by vacuolar fusion within cells and then vacuolar fusion across cells to give rise to these intracellular luminized intersegmental vessels. So it's a very simple system where you can ask which genes control the branching processes, how is lumen made, and so on. You can ask, do cells rearrange? Do they rearrange the same way they do in the flies? Are forces distributed the same way along the system? How do vessels fuse? There is nothing known about vessel fusion. There is not a single paper on how blood vessels fuse. Yet there are 15,000 papers about VGF, who is sort of the ligand that drives formation of these sprouts. So let me give you one brief example now of how we started to look at these questions again at the cellular level, because we think what cells do is the key point. The junctional arrangement of these cellular arrangements here have not been looked at in zebrafish because there are no antibodies available, more or less. So the model came from single-cell labeling via transient expression of plasmids. But when we looked at these structures, it was quite obvious that there were various ways you could put in junctions here. And the way you put in junctions might actually impact the way these tubes form and the way the angiogenesis process proceeds. So you could think that this cell here is a cell that has 2 neighbors, like in the trachea, and has actually an autocellular junction. So then it could arise the same way that we see these cells arise in flies. They are adjacent, they intercalate, and so on. But it could equally be possible, because it has been proposed that these are intercellular lumens, that you have only small rings that connect this cell to this cell. These are endothelial cells, which have adherence junctions. So you could have either small rings and lines, and then things become complicated when you come to this cell here, which is a cell that has no lumen at first, and then lumen must be generated to interconnect. So there are various ways these cells could organize, and when we then used antibodies, and we also generated our own antibodies against weak adherin, we realized that the junctional arrangement that we saw, mostly 2 lines, up here, down here, and very complicated up here, was not consistent with the cellular architecture as it was proposed by these preliminary studies from Mark Fishman. So there was something going on that we didn't really understand when we looked at the weak adherin antibody, which is shown here. This is a SEDO1 antibody. You can see that the SEDO1 staining, which also stains other things, completely overlaps with the weak adherin staining. So the 2 lines we see, we also see them with weak adherin, which is exclusively made by endothelial cells. So clearly from these junctional stainings, we would argue that the cellular architecture of these vessels is different. Cells are actually paired in these branches. The lumen is extracellular, not intracellular. So the way these branching process actually happens at the cellular level must be re-examined. But before we wanted to re-examine how such branches would form, we of course again wanted to be sure that this model is correct by doing single cell labeling. So exactly the same approach as we used in flies. If we would take this single cell approach and confirm the model before we make any model about any mistake about the model, and then actually try to explain things that don't occur. So if you would label single endothelial cells, this model would predict here that these single endothelial cells have junctions all around, and at the place where two other cells meet here, you might find additional lines which do not belong to these here. And this was actually confirmed. So the model can be confirmed with single cell labeling and junctional labeling. You can also confirm the model by labeling all the cells in one color and individual cells in a single color, and ask whether these cells are actually throughout the lumen or whether they actually pair. And you can see that these single cells here nicely pair along the axis as they would in this particular model here. So it's quite clear that the model argues that the architecture of these branches is an architecture like this, paired cells, and we have to ask now how these branches then develop. Do cells divide? Why do we see all these kinds of junctional lines up here? Do cells intercalate as they do in the tracheal system? And obviously, again, you would like to use live imaging approaches for this. So what we first asked is, do cells actually divide during this branching process? And I will skip over the movies because it's easier to show in stills. So what you can see here is a nuclear marker that goes to the cytoplasm as the cell divides, and then back into to the nucleus here. This is a stalk cell that divides, and slightly later the tip cell also divides to give rise to two cells. So there is extensive cell division as these branches grow out. And we also label single cells to ask whether there is extensive cell rearrangement. And here you can see that cells born in the stalk, they actually can move up to the ISV as development proceeds. And sometimes these stalk cells, they even go over here and enter the stalk of another cell. So the whole process is extremely dynamic. It would virtually be impossible to come up with a solution by using fixed tissue. Cells divide, they reorganize. A lumen has to be formed. Lumen has to be formed in tip cells and so on. So the process remains sort of obscure. And I think the best way to attack it is to really go on with live imaging until you describe the process. I just would like to give one more example now of how we do that. When we ask how is this cell, which is a tip cell, similar to a tracheal cell, actually opens up the lumen with its neighbors here on either side to give rise to these interconnected ISVs. Can we learn something from flies? Flies also fuse their tracheal metameres, and they do so by basal membrane that contact between two different metamers contact each other. An ecadherins spot is made, and between this ecadherins spot and the pre-existing apical site, a tunnel is dug through the cell, and you generate cells with two apical sites, two cells with two apical sites that lead to this particular fusion process. The lumen is intracellular here. You can see how that goes in flies. So you have two adjacent cells here that come together. They generate a spot of ecadherin. The tunnel is made now between these sides here. So we know quite a bit in the tracheal system how that goes. Fusion cells in the flies can be recognized because they have these specific they are donut cells. They express specific proteins. So the first question we ask, can we recognize fusion cells in the zebrafish embryo? And the answer is no, because when we look at the junctional arrangement here, nobody could derive from this that here, yes, there is a fusion point. It could be here. The junctional rearrangement looks exactly the same. So how does this cell here, the tip cell, which connects its neighbors, then open up this lumen here? It's not such an easy question, but what we can see is that there are some similarities with how flies do it. Tip cell philopodia from these two fusion cells, they meet each other. They generate a first line of ecadherin and junctional markers, and somehow then the lumen is made between these two apical sides here. When we look in still pictures, what we could think is, yes, there are first contacts here where de novo junctions are made. These junctions then somehow elaborate. They become bigger. Cells crawl over each other, in contrast to tracheal cells that actually intercalate. Here we have somehow cell crawling over each other, but this obviously doesn't explain how the lumen from here opens up to the primitive de novo lumen shown up here. So based on some of these studies that I've shown you now, we propose the following model. We propose a model for vessel fusion which is actually quite elegant, because at the end of the day, the vessel has exactly the same cellular architecture in a fusion point and in a non-fusion point. It remains very flexible. What cells actually might do is contact each other, elaborate a new apical site extracellularly, and when some of these junction points here meet the next junction point here, the cells can rearrange so that the lumen opens with any membrane fusion. So this is a model based on some of the data we have. Very different from tracheal cell fusion. The lumen is extracellular, cells only rearrange. There is no membrane fusion events to open up the luminal space. To finish, I would just like to mention that such a model for fusion has been proposed. It's virtually identical, this model here, from work done on Siona intestinalis, notochord, where people showed that complex cell rearrangements are responsible for lumen formation in the notochord. And when you look at these things here, they're complicated to view. They actually are in line with what we would propose for blood vessels. But how can we, again, now confirm this model? How could we show that this model, compared to other models, is the prevailing situation? I think what we should do, again, is use live imaging and use single-cell tracing. So we have generated fish that make a usable form of GAL4 and can cross these fish to fish that make US Caide protein that can be induced, or the green protein can maybe made to a red protein, and we can track these singly photoactivated cells now during the fusion process and ask whether the lumen is intracellular, extracellular, how the cells actually behave. But what we really wanted to do is to look at junctions, because with junction you can see what happens. And I'm quite excited to show you now the first movie that we did last week. We have tried many times to take a flea promoter and hook it up to Z01, to alpha-catenin GFP, to any junctional marker. And years of work were giving nothing, because either these fish survived and the GFP was not strong enough, or they didn't survive. So we never made a line that was usable. So we tried to go to the US GAL4 system, made flea GAL4 fish and US Z01 fish. And when you look now at these heterozygous transgenic animals, you can see this is a tip cell here. The tip cell must have just contacted the other tip cell from the other side, and we can simply ask now, and I don't have the answer to that, unfortunately, what happens to this spot and what happens to the lumen as it opens? Does this spot refine to become a loop and a bigger loop and meet this junction here and then only open up, or what actually happens? And the movie is too short, so we have to make many more movies, but the protein is very strongly expressed. You can see what actually does happen is, yes, this spot refines to a larger ring, and we have to make now more movies, and these movies will show us how this lumen then actually opens up. Clearly, the GFP protein overlaps completely with the endogenous protein, so we are sure what we are looking at is actually relevant. So we have to do a little bit more live imaging until we understand the cellular basis for this ISV branching morphogenesis and understand lumen formation, how is it correlated with cell migration, cell division, cell rearrangements, and so on. So I think the system that we might eventually come up with might or might not look very similar to the tracheal branching morphogenesis process. Clearly, the two have key molecules that control key aspects. It turns out that VEGF also controls delta, controls tip cell formation. It also controls cell division, which is not the case here. So I think models are now emerging where we can ask what is similar and what is not so similar within these two branching morphogenesis process. If we go to other systems, the situation becomes very complicated, because there are genetic regulatory networks that are involved in limb development, lung development, kidney, or mammary gland development. But the cellular basis in all of these systems is either completely unknown or hardly explored. What do these molecules? Some systems have molecules that are locally expressed in the mesenchyme in front of the tip? Do they control cell division? Do they control cell migration? Are they chemotactic substances and so on? The underlying cellular basis is completely unclear because there is no way to film lung development in an intact animal. There are limited ways to film kidney development. Limb development, obviously, would have to be filmed for many, many hours, and that hasn't been really tried either. So I think it will be very important to link these networks to the underlying cell behaviors, and then one can ask what is similar or what is not similar, and then maybe appreciate a similar or not so similar cellular and genetic control of branching morphogenesis. So with this, I would like to stop. I would like to thank the people who have either done the fish work or the fly work, and the people who provided the money. And thank you for your attention.
Mark,
Yeah.
microphones, please.
You can speak loud.
Yeah.
Yeah.
If you ever look at the intracellular structure, that is, particularly the cytoskeleton, I mean, for example, is there some polarity to the cells? Are there some lamellipodia or filopodia or things that would give you a sense of the underlying process that's driving the morphogenesis?
Yeah, we didn't look very much at that either. Obviously, we have GFP-labeled tubulin, but one should have GFP-labeled centrosomes and so on, and we have not very much explored these things, but that should obviously be done. That's clear.
Does tubulin evolve? I mean, do these things form in the presence of the cosmos?
No, tubulin is of course these microtubules are very important for these structures to form. That's very clear. They're not very dynamic, obviously. So we used here the actin. We mostly used it to track cells that are actually dynamically remodeling actin as being those that actively migrate. So we used it more as a readout than as anything else, and that's why we didn't look at tubulin. It's very difficult, and we started to do genetic screens to go into the cell and find out more about what happens actually in the cell. What is downstream of FGF that makes FGF signaling more prominent, more eager to be linked to such a system than EGF signaling, because we replaced the intracellular domain of the receptors and so on. So FGF is intimately linked to that. EGF cannot do that, but we don't really know how this connects intracellularly. So there is something specific about FGF signaling, that FGF signaling can do much better than EGF. Is it duration? Is it localization of components? We don't really know. And it's very difficult to do genetic screens to find that out in a multicellular animal. So we need to go to other cell systems, RNAi, tracheal cells and culture and RNAi, to find out more about the intracellular behavior.
Yeah, I have a question regarding the mechanical stimuli or the power that drives the crawling of the cells. Is it only the tip cell that just pulls them, or is it something inherent to the cells themselves that...
No, we did quite a big series of laser ablation studies to ask whether cells do this intercalation process, do they actually generate their own force? Is the tip cell force the one that guides the other processes? And it's clearly tip cell migration that is sort of dominant over any other force generation in stalk cells. So as soon as you unlink the stalk from some other cells and the tip cell force can not exert its force anymore, then the cells don't intercalate. They're completely non-motile. But as soon as you remove tip cells, stalk cells become tip cells. So there is I think the key also to the other branching processes is how to generate a tip that is distinct from a stalk, and then how to make the tip behave the way it should behave. The stalk is usually not the problem. Branching morphogenesis is a problem, how to generate tip, how to distinguish tip from stalk. And once you have generated this, which might be through cell competition, then you have a structure that can sort of give rise to branches, and then you have to deal with where is the next branching point.
Mm-hmm. With regard to that, can you comment anything about the Malpighian tubules?
There is the Malpighian tubules. There is very little known of how they disperse in the embryo. There are also chemotactic signals to that, but they have sort of a mitogenic signal on top, which allows the structure to grow and sort of branch because of local cell division. And these are sort of the questions that have to be asked in lung, kidney, mammary gland. I think each of them has its own system to do it, which actually is clear for trachea and the vasculature. You have to reach every point in the embryo. And you do that with hypoxia control, as we heard, so they're very elegant systems to regulate that. The lung doesn't want to reach all organs in the, it branches in a bag. And the mammary gland does it maybe in a stochastic way, while Mark Krasnow has just shown that the lung does it in a very genetically hardwired way. So it will be interesting to find maybe common cores, tip stalk generators, and then you can control this maybe in environmentally or not environmentally controlled. But there is too little data at the moment to really ask many questions.
Okay, thanks.
I have a question. So, in other systems, for example, in filamentous fungi, in Neurospora, and in sea urchin, and also in C. elegans, where there is cell-cell fusion in branching morphogenesis. However, you've never seen this, not in zebrafish and not in Drosophila.
No.
You think that you may have missed, and that in some cases, either the cells can fuse with themselves, the actual membrane fusion, or that you get actually two tubes or two rings fusing?
I don't think that cells fuse. They're multinucleate. I think cells don't even, if they don't even fuse in Hüvec cells, then they will probably not fuse in vivo. Hüvec cells do a lot of crazy things that people have thought that they would also do it in vivo endothelial cells. But I think not even in Hüvec cells these cells fuse. So if Hüvec cells don't fuse, I think there will be no fusion in vivo, where the whole Hüvec cell behavior is actually under very tight control. So I don't think, we didn't see it, nobody else saw it so far. There is obviously membrane fusion will occur, but between apical membrane and apical membrane vessels, things like that.
Okay. Are there any other questions? Okay, so thank you very much.